Hello everybody and welcome back to the Sanctuary. I'm your host, Professor C, and we're going to dive deeper into the world of RMP, specifically into the world of muscles. We're going to check out membrane potentials, specifically the resting membrane potential and the generation of an action potential. So let's see it. Alright, this talk can be challenging for a lot of people, so I'm going to try to keep it as simple as I can and get to the point as quickly as I can. So what I'm seeing here in this first image is a cell membrane. I can see the phospholipid bilayer in the background. And then I see a whole bunch of these cylinders and ovals and arrows, and we'll get to that in just a moment. The first statement I need to make is that right now we're looking at a membrane at rest. And we're going to introduce this topic of what's called the resting membrane potential. So there exists an uneven distribution of charges on the inside, intracellular, that would be right here. We'll call this the ICF, the intracellular fluid. You could also call this the cytoplasm if you wanted to. And the extracellular surface, which would be out here, this would be the ECF, the extracellular fluid. There are other things you can call it as well. I'll be referring to them as ICF and ECF when I mention them. So what we usually see is on the outside, we see a lot of sodium ions, and I'll just draw it like a big ball like that. And wherever you see sodium in biology, you're likely to see his little pal chloride, right? Because it makes salt. So we're going to see an abundance of sodium and chloride in the ECF. And we'll see a tiny little amount of potassium. So I'm just going to put a K plus here to represent the tiny little amount of potassium. Now on the ICF it's different, and remember we talked a long time ago about how cells want their internal environment to be different from the external environment to maintain an internal homeostasis. So on the inside we see something different. On the inside we see it dominated by potassium ions, lots of K ions, potassium on the inside. We also see a lot of protein, and I'm going to write the protein as amino acid because sometimes it's just strings of amino acids, and they're very heavily charged negative. I'm also on the inside going to see a little bit of sodium, not very much, a little bit. I'm going to see a little bit of phosphate, PO4, a little bit of that. Notice the negative charge. I'm going to see, of course, if sodium is playing, I'm going to see his little friend chloride, but just a small amount relative to the rest. So the dominating players on the ECF are sodium and chloride. The dominating players on the ICF is potassium and protein. And so we get this uneven distribution of electrical charges on the inside and outside surfaces. Now these are regulated, maintained at rest by what we're calling them the blue channels are called passive leakage, also the green guys, and these are, as it states, passive. Now the blue ones are K channels that leak out. So there's a K channel, there's a K channel, there's a K channel. Now these are just like holes in the membrane. All the blue ones that you see the arrows going out are passively leaking potassium ions from the inside to the outside. So we say K out, right? Potassium is being pushed out. And notice how many there are. There are a lot of those blue cylinders and arrows pushing passively potassium ions out. Now right next to it in green is another passive channel, but they're pushing sodium in. Now there are many fewer sodium channels that are passively letting sodium in than there are passive potassium channels letting potassium out. Now, there's another way this is regulated as well. It's the active pumping of these oval-looking pumps. And if I can give a little ATP to these pumps, and I'll put a little flash around it so we know that ATP is a powerful, kind of a powering type of molecule. So this is going to push 3 sodiums out and bring in 2 potassiums in. And remember, we can do this, we can push 3 out and pump 2 potassium in, even though that the sodium is already abundant outside and already the potassium is abundant inside, because we're using a powered active pump. Now, clearing everything off, we can make a few more statements. That sodium is dominating the outside world, and that potassium ions are dominating the inside world. Just to remember, this is the ICF, just to keep it straight, and this is the ECF out here. Now, because there are so many amino acids / proteins and phosphates down here, that actually gives the inside surface a negative charge. And it says right here, there's negative charges along the inner surface. I'm just going to draw them just like this, little minuses all the way across, to show us that the intracellular surface is negatively charged. Now, because sodium dominates the outside world, the outside of the cell has a positive charge to it, just like this. And I can go all the way across. And again, we looked how this is maintained, but these charges are separated in space, right? There's a cell membrane separating this relative positive charge that's outside, this relative negative charge that's inside. We use the term polarized to describe this, just like a magnet would have a plus pole and a minus pole. Opposite charges do attract, however, and those charges are trying to get with each other and cancel each other out, if you want to think of it that way. But there's a membrane in the way. And so there is energy required to separate those charges. We call that energy a voltage. Now, the force of that voltage has a potential that gives us a potential to do work. And right now it's not doing any work because it's resting. So that's why we get this concept known as the resting membrane potential. So it got the potential to do work. However, at this moment it's not. It's just resting. But it could do work if we were able to get it active somehow. So it says here at rest, which we're looking at, there is a different kind of channel that I didn't show in that picture called a voltage gated channel. And it looks like this picture here, which is a very nice one, that shows kind of a ball here and a chain hooked up to it. And it's kind of blocking this path. And if it's hit, if you want to use that term, with some stimulus, if we can change the voltage a little, if we can make the voltage less negative, okay, if we can make the voltage less negative, that ball and chain will move out of the way, and sodium ions can rush into the cell freely. But right now it can't because the channels are closed at rest. We have established then this resting membrane potential. And if I could put a voltmeter, some sort of tool, a device that measures voltage, and I could jab it into a cell membrane just like this, and has been done many times in many labs, it would read negative because remember, it's more negative on the inside, on the ICF side, than it is on the ECF side, which is more positive. Notice they're calling it cytosol. It's another term for the cytoplasm. The number is -70, and it's recorded in volts, but these are very tiny, tiny fractions of volts, thousands of volts, so we're going to call it 70 millivolts. -70 millivolts will be a great resting membrane potential. So in this chart, we can actually show that. Look here, and we can say this axis here is millivolts and will always be on every chart I show here, and time will be here on this axis. It's milliseconds is usually what we measure that in, but I'm not going to put specific timing with this. So if we were going to, say, measure this millivoltage with a voltmeter, we would find that a normal neuronal resting potential, and it's right here, they got the British spelling for neuron, but that's okay, is 70. Notice it's different in other cells. But we're looking specifically at a membrane potential of a motor neuron that's going to try to turn on a muscle fiber. If we were to graph out a cell, a resting membrane potential, well, we're done. I can put RMP to show that. It would just be a flat line at -70 millivolts for all the reasons we established on the first couple of slides. Now, let's say we want it to do something. Let's say we want the neuron to wake up, right? We have to wake up this motor neuron in step 1 here of excitation-contraction coupling, and we're going to do that by opening these voltage-gated sodium channels. Remember, they stayed closed. We had some leakage channels, we had some active pumping, but now if we hit this membrane with some stimulation, we can get those ball and chains to move out of the way, and what we'll see is an influx of sodium. And what happens, as I said, we're going to have to get this to become less negative, right? So what we're doing is we're trying to move that voltage towards some certain number, and it's called a threshold. And for a lot of the neurons published in the books, it's somewhere, threshold is, I'll just put thresh, is somewhere about negative 55-ish. Okay, sometimes negative 50, it's around 55. So there's a magic bar here somewhere that's usually shown as a dashed line on many of the textbooks right there. So what we're saying is, if we're at rest, and we know that's negative 70 millivolts, if we hit this neuron with some stimulation right there at this time right there, we're going to see sodium come in, and that's going to make the potential less negative. That means we're going to see it blip up like this. And if it's not a very strong stimulus, it's going to blip right back down to resting. Now this is a depolarization, but it's not a very strong one. As we said, it didn't hit this critical threshold number yet. So this is known as a local or weak depolarization because it did depolarize. It started to wake up, much like some of you might wake up in the morning, but it immediately hit the snooze button and fell right back to sleep. The stimulus wasn't intense enough. So we moved the voltage, we did a weak depolarization, but it didn't wake up the neuron all the way, meaning we didn't hit threshold. So nothing happened and it went back to sleep. So the notes here reiterate that. If the stimulus remains sub-threshold, like we just showed, it will return to rest, and nothing will occur as far as making the neuron get excited. Sometimes, however, you have to get not just one potential, but you have to get multiple weak potentials and add them together, like the sum in an arithmetic class. You must do summation. That is something like this. Remember, here's threshold around negative 55-ish. Here's a membrane at rest at negative 70. Let's hit it with a stimulation. Oh, it wasn't that great, right? We definitely hit it. Sodium channels open. Some sodium came in. We had a local depolarization, but we didn't hit threshold. So what happens, if we hit with another stimulus and another stimulus all at the same time? Well, you notice those are adding together, and they're getting us closer and closer to that threshold. They're making it less and less negative as we go, or more positive, you could argue, because sodium is coming in. And this is always caused, depolarizations are always caused by sodium leaking into a cell. Okay, so what happens if we actually get enough weak potentials to add together where they bounce against that threshold limit here? Well, we're going to actually wake up. We're going to go full-blown action now. We're not going to be in a resting state. We're going to go into an action state, and we're going to form what's called an action potential. So at threshold, which again is somewhere around negative 50, 55, somewhere like that. At threshold, if we can get okay, here's rest, here's our local or weak depolarization. If we can get it to hit threshold, we're going to see a lot of voltage-gated channels open, and you see that over here on this side. So the ball and chain is moved out of the way, and you're going to get essentially a flood of sodium coming into the cell. You get a positive feedback loop, and this will increase the membrane's permeability to sodium by about 1,000 times. So this is usually called a flood of sodium, even though it's not quite as pretty as you might think in your mind. It certainly works for passing the test, right? An action potential is formed, and this is generated as what we call an all or none phenomenon. So what happens is I get a spike up here very rapidly, and this is our action potential, and it usually crests out about plus 30 on almost all the textbooks that you'll see. It's almost a 100 millivolt reversal from its resting at negative 70 to the spike of the action potential at plus 30. It's about 100 millivolts. So remember, this is caused by sodium coming into a cell, whether it's here weakly coming in or whether it's here flooding into the cell. The depolarization, and I can put dipole just so we all see it, the depolarizing wave is caused by sodium coming into the cell. And we get an action potential, a wave essentially generated of a magnitude of plus 30. Now all or none is just simply stating, If we hit threshold, we will form a plus 30 action potential. If we don't hit threshold, we will form no action potential at all. You won't see a short action potential that goes something like this, right? It won't do that. It will either go all the way to plus 30 or it will not form at all. And this is called the all or none phenomenon. Okay, well, the membrane doesn't like that, much in the way that you don't like being woken up by your alarm clock. So if I could draw an axon of a motor neuron, something like this, what we've done is, remember, it used to be plus on the outside, and I can kind of show it just like this, and minus on the inside, just like this. But when we hit this little area with stimulation, everything flipped. And there's our depolarization event right there. As that sodium flooded in, we had a flip or a reversal because it went into the positive numbers, right? So the membrane doesn't like that. And it will want to repolarize itself very quickly. And it does. So it's going to slam channels shut as soon as it hits this action potential. And it's going to start continually spewing out potassium now. So potassium is going to exit the cell freely. Some pumping will happen. Some passive channels will spew it out. And this is going to restore the polarity back down toward rest. So let's check it out. Here's our resting potential. Here's our threshold level, somewhere about 55. And now we're going to hit it with a stimulus that is above threshold. So we get a weak or local depolarization. It's going to snap up as an all or none phenomenon into a full-blown action potential. Remember, this is caused by sodium coming in. And this is the depolarization phase. Now what we want to do on the other side, as this slide is telling us, is we want to repolarize. We want to come back down. So this is the repolarizing phase. And that's going to be caused not by sodium being kicked out, but by potassium being kicked out. And the word for kicked out is efflux versus influx coming in. And that's simply what's happening here with this downward stroke of this on this graph. Okay, now during this time, and it's somewhere, it shows it differently in different textbooks, but somewhere from about here to about here-ish. There is a time in here where you cannot stimulate this fiber again because it's already been stimulated. This is called a refractory period, the area I've drawn in green, and the one I've drawn specifically is called the absolute refractory period, where you absolutely cannot stimulate that neuron again. Okay, well, that's not the end of the story, actually. What we have happen is, again, there's my threshold level. Here's resting. I'm going to hit it with a weak or local depolarization, then go full-blown flood of sodium in, positive feedback, to form the all or none action potential. It will then start to drop because of repolarization. Just to remind you, this is sodium in. It's causing this upward slope. And this is potassium out, causing this downward slope of repolarization. But it doesn't stop at resting like you would think. It actually continues down, further negative, and then it will come back up and go to rest. And this little event here where we overshoot our target is called hyperpolarization. Now, the point of the hyperpolarization is to extend the refractory period. Remember, we had somewhere in this area an absolute refractory period where we simply could not stimulate that neuron again. And so when we're in this hyperpolarization phase, there's a second or an additional refractory period that lasts all the way until we get back to a nice, beautiful resting state somewhere over here called the relative refractory period in blue. And here you can stimulate that neuron again, but it's got to be a very strong stimulus simply because we are further away from resting than we initially started. And then we'll come back to rest. So this hyperpolarization is where we pump a lot, and I can put like excess K out. And that will undershoot the target. Again, extending the refractory period and making sure we don't overstimulate this particular structure. Once we've done that, and again, I can draw a little axon here. Remember, we hit it just at one little area with the stimulus, and we flipped those charges, both plus to minus and then minus to plus, and that caused kind of a ripple, as you see there in my water picture on the left. and we drop the pebble at this one spot, we don't have to hit this axon or this sheet of water with more stimulus. One pebble was enough. And so a depolarizing wave spreads across the axon to adjacent areas. When this depolarizing wave hits the adjacent areas, sodium channels open and we have deja vu. We have the same thing happen again. It will cause dipole, repole, and then the next adjacent area will depolarize and repolarize. And we get this kind of flipping of light switches, almost like a domino effect that you see on the right, where we just have to form one single event and then the rest can happen just naturally. Now, where we're pushing this action potential is toward the terminal knob of the axon, or we could say the axon of the motor neuron. And waiting on the other side would be a muscle fiber ready to be stimulated. So this is the excitation part of the excitation-contraction coupling. We have to finish the story in two more different short stories. We have to first jump across the gap and then stimulate the muscle. Conduction velocity is just a term that relates to how quickly those action potentials are propagated. And again, that's that fancy word for pushing them down the axon, propagation. How fast they move is dependent on a couple of factors. The main ones you see in text are the diameter of the axon. And the wider it is, so the higher or bigger the diameter, the faster the conduction velocity, which kind of makes sense. A big pipe would allow you to have more water flow through it. It's a very similar idea. So the bigger the diameter, more the conduction velocity. Now, myelination is not something we'll talk about during this lecture, but there are sometimes some insulating structures that wrap around axons. And if we do have myelination, the more myelination we have, the faster conduction velocity we have, because it will travel differently in a saltatory type of conduction that we'll talk about in the nervous system chapter. All right, just to do a real quick review, here you see on the picture on the right is a very typical image of a membrane going from rest, forming an action potential, and then coming back down to rest. So the red line, which is right here, would represent resting membrane potential, and again, that should be somewhere about negative 70 millivolts on most of the charts. Again, somewhere around 55-ish, and it's different everywhere you look. It's somewhere between 50, 55. We need to hit a threshold number. Now, event number 1 here looks like something happened. It woke up a little bit, but it didn't actually hit threshold. It was just a sub-threshold depolarization. We can say that's like a sodium leak in, just a little bit. Now, if we have multiple subthreshold stimuli, we can summate them up to threshold. So at threshold, we have a threshold depolarization. That's where the voltage-gated channels open, and that's where we get this flood of sodium come in. And that produces this upward spike of the action potential. So remember, sodium in, just to beat a dead horse here, but sodium in is what causes this depolarizing phase. The action potential is reached at the top, and remember, all action potentials are going to be the same. This should be plus 30 if we're talking about this neuronal system. And then very quickly, this neuron wants to go back to sleep. So it will start a repolarization phase, and to do that, it will not pump the sodium out. It will instead close the sodium channels and pump out potassium ions as fast as it can. And it doesn't stop when we hit resting, right? It actually continues. There's an excess amount of potassium pushed out. This is known as hyperpolarization, and that is, of course, to increase the refractory period from an absolute to a relative, making it harder to stimulate the neuron for a period of time. Once it full-blown goes back to rest again, you can start the slide over, and you're back to where you started at resting membrane potential. Okay, that was a complicated one, but thanks for watching it. Check out some other videos in the series if you want to learn more about muscle physiology and anatomy. Bye-bye.